I'm here. Yeah, I'm here, guys.
Here?
We were just in a different meeting. The ABL people.
Hey,
Good morning.
Aris, how you
Hi,
doing?
everybody. So yeah, so Reza is going to talk to us today about the platform he's been developing for the last couple of years. Should also mention that Reza is the assistant director of ABL. He has done some great work for many groups here at NYU, but I'm going to let him speak and looking forward to the talk, Reza.
Thank you. Let me just share my screen then and start. Okay, share the screen.
And I want to give a shout out to our cancer director, Ben Neal. Hey, Ben.
Okay. Do I make this small? Yes. Okay. Does everybody see the screen, right?
Yeah, perfect.
Okay. So let me talk about UMAP. This is a dimensionality sort of dimensionality reduction that we've developed in fact a few months ago. It's unpublished. It's actually the first time we're publicly presenting it, but some labs have used it, and I guess like 30, 50 projects we have been using it for the last few months. So today I'm going to be actually comparing it to tSNE and UMAP and diffusion maps such as Destiny and PHATE. Just before we start NetL map, the K is not pronounced like words like knee, knife. So it's like the plant NetL, the wheat plant. So it has that leaf logo on it. So before we start, does my slide change or not? Let me see. Okay. Oh yeah, it does. Okay. So before we start, NetL map is available online. It's open source. You can install through CRAN or GitHub. Also, there's plugins for it from Flojo or SeatGeek if you're using those. In fact, is one of the more popular plugins in Flojo and SeatGeek. But so the pipeline very quickly, it looks like most of the single cell pipelines. So you get your data, you make an object, you filter them based on quality of coverage and mitochondrial or any other parameters you want to normalize with whatever method you like to, and then select genes of interest for supervised or unsupervised clustering, and then run PCA or batch alignment. In iCellR, if you run batch alignment, it automatically also runs the PCA, so it replaces that. So either have to run PCA or batch alignment. You have 4 for batch alignment. You have 2 methods that we have developed. So one is called joint or combined PCA, CPCA and CCCA, another method. And there's the other two. There are 2 wrappers for if you want to run mutual nearest neighbor or multi-CCA. So then after doing that, you run either tSNE, UMAP, very commonly used, or some people do some diffusion maps such as Destiny and PHATE. In here, we're going to run NetL map as well and compare it to them. And then the rest is clustering and finding markers and the downstream analysis. So mainly today I would be testing NetL map with these 4 methods. One would be comparing it to in terms of time and performance, and to tSNE, UMAP, and PHATE and Destiny. And then we would have a complex data set such as mouse cell atlas or human cell atlas, and compare it there, as well as low complex data such as mass cytometry, which has only 40 proteins and antibodies. So it's less complex than like 25,000, 30,000 genes. And then we would also have it compared to data sets that are very hard to cluster, like dendritic cells or T cells that are like usually come as one big blob in UMAP and tSNE, and see how NetL map performs in there. In fact, Joseph might have slides because they have some really experience on dendritic cells. And then also compare NetL map on data that is batch aligned. So just a quick, just one slide on the algorithm, how it works, and then we would dive into the comparisons. So so NetL map is actually based on networks or graphs, And networks are simply put, there's just two things. You have two things in a network. You have nodes and links. This first paragraph is just saying that to make a graph or a network, you just need a node, nodes and links that connect them. And these links or edges could be directional or unidirectional. And so in NetLMap, what we're using is actually we're trying to draw these networks. And the method that we use for drawing or layouts, also called layouts, graph drawing layouts, is a force-based layout. And these layouts usually they have, they define them as having two forces: one attractive force, which is they usually call it analogous to spring force, and the other force is the repulsive force, which they usually define it as electrically charged particles, like in atomic particles, kind of like so. So this first was developed in 1991 by Futterman and Reingold. So the way NetLMap works is this diagram. It shows the whole process and down here. So what we're doing is we're really playing with these forces to such a point that we are making the cells, by playing the force in the system, you're making the cells really so compact that they all squish into these compact nodes that they look like there's only one dot. There's thousands of cells here, but they only look like there is one dot. And then we extract the dimensions of the nodes, and then we release and we unpack them. We open them up a little bit. And that's how NetLMap works. Here's the first comparison. This is a very commonly analyzed data set. First was put up by 10x Genomics for people to run as a sample data set for testing out Cell Ranger and some tools. So this is a PBMC data set. You have this cluster in tSNE, you can see here, cluster 10, 9, 6, and 7. Those are T cells. And then you have some myeloid cells here, 3, 4, and 5. And there's B cell and natural killer cell in the bottom here. So as you can see in the, compared to PCA, UMAP and tSNE, you can see a little bit better separation among the clusters. So that's why tSNE and UMAP are more commonly used in single cell, because they perform better in terms of visualizing. And in PCA, usually like you have many dimensions. The first and second one are showing the most differences. Whereas in UMAP and tSNE, you generally like trying to squeeze some of those dimensions into one or two. So you get a little bit more clarity. So again, and here you have the Destiny and PHATE. These are diffusion sorts of dimensionality reductions or diffusion maps. As you can see in the tSNE and UMAP, cluster 9 and 10 and 6 and 7, they're very close to each other. And it's kind of sometimes arbitrary to, like to draw the line to really like get them with more distinction. Whereas in NetLMap, which is the last one in here, you can see all of these clusters are having clear boundaries, and they're like really defined in terms of look. One thing about NetLMap is that it doesn't keep as much global structure like UMAP does. So global structure and local structure, usually people use it. It's kind of like a pseudotime thing. So if the closer the clusters they are to each other, the more related they are to each other, right? So in UMAP, the tSNE keeps a little bit of that, less of that. UMAP keeps more of that global structure, but NetLMap unfortunately doesn't. And that's because NetLMap tries to fit as many as the cell communities in one slide, but all of them with clear distinction. So you can see all of them in separate spaces. And another thing about NetLMap is that it has a zoom option. So you can zoom in and out. So if you zoom in more, you would see, for example, this cluster number 9 would be dividing into more subpopulations. and more of these clusters. So you would see, if you zoom out, you zoom in, you would see more and more clusters. As you zoom out, it would look more and more like tSNE and UMAP. So because the NetL map doesn't keep the global structure, we have a abstract version of what you see in the right, these two plots in here. Those are abstract NetL maps that they're trying to show you which cluster are more closely related to each other. They look more similar. So, and then in the top right, you see the time comparison. Here's the number of cells, 1,000 cells, 2,500, and all the way to 50,000. You see generally, you know, tSNE, UMAP, and PCH is generally up to 50,000. They're below around 10 minutes. Whereas in NetL map, Destiny actually goes a little bit higher. But NetL map, depending on the number of the amount of zoom you're using, the time increases. So around 50,000 cells, you're looking at about, you know, 2 hours or 1 hour and a half, depending on your processing power as well. So it takes a little longer. However, you know, imagine if you have large projects such as, you know, mouse cell atlas, where most of the people have, you know, spent so much time to do broad clustering to define the clusters. It would be helping you a lot there. I would show you a couple of slides that, and then it would reduce the time in that sense. Time analysis. So here, so in the next slide, so here we have the NetL map. I wanted to show you some genes, turn them on the NetL map, and so show you how do they look like and how you can define the clusters in there. So here we have two versions of the same NetL map. One of them is more, you know, we did more unpacking, so the clusters are a little bit not as squished. So they're a little bit more open on the right, and there is the original one on the left. Usually the default and the original one looks good, but there is a little bit of like, you know, might need a little bit sometimes unpacking. So anyway, so here you see, if you turn on CD3E, you see these 4 clusters are T cells, and of those, 2 of them are CD8 positive, and actually in here, maybe on the right would be easier to see. So again, so cluster CD3E. So these 4 clusters are CD3E or T cells. And then we turn on CD8. You see these 2 are CD8 positive. One of those CD8 positive is GZMH positive as a marker. GZMH is also sometimes comes up high in natural killer cells. You turn on KNLY and it turns on in here. So that's the bottom one is your natural killer cell. And it's negative for CD8, as you can see in here. And of the other 2 T cells, if you turn on CD4, CD4 also comes up in myeloid lineage as well. So these 2 are your CD4 positive. CD68, these would be your myeloid lineage. Of the 2 CD4 positive, if you turn on CCR7, it comes more in this one. So that would be your memory T4, and then this would be your naive T4. Again, and then if you turn on FCGR3A or CD16 to distinguish between your myeloid lineage, you see FCGRA comes highest in here. It's also coming high in natural killer cells, but among the myeloid lineage, it comes up here. And if you turn on CD14, where that would be CD14, yeah, these 2 are coming as CD14 positive monocytes. So this one in the middle and down here, that would be CD16 positive monocyte. MS4A1 or CD19 would be your B cell. As you can see, this long one is in here, and the B cell, and in the left, that would be this bottom one in here. And so as you can see, it's easily, the markers can come and you can distinguish the cell types in there. So here is the same clusters. I put in a zoom of 50 and a 200 for you to see. So then the lower numbers, you actually zoom in. The higher numbers, this is kind of reverse. The higher numbers, you zoom out. So as you can see, this 8 that with the zoom of 100 was about to split, but when you zoom in at about 50, they split easily. And then that's 2 types of B cell. And that depends, the zoom, it really depends on how much detail you want to see and what you want to see. And actually today, mainly I would try to talk about how to decide what zoom is best and how to adjust NetL map, because unlike other dimensionality reductions, this one needs a little bit of adjustment and knowledge of your data. So, and this is the 200, as you can see, the 9 and 10, they can't come closer together. 6 comes in the corner here. It looks like more tSNE and UMAP, this corner, turn on gene. So it's not like as separated with clear boundaries. So, and the 4 and 5, they're similar colors, but they're actually on 2 different sides of this one. The shade of color is kind of similar in there. Okay, so that's how you zoom in and out. This is another slide working on mouse cell atlas. Here on the left, you see a tSNE and a UMAP of the same data and a NetL map in here. There's 20 tissues on this. Here's the data we downloaded. We have actually downloaded a few other versions of mouse cell atlas as well. In this one, there's 20 tissues in here. As you can see in the tSNE, it finds about 20 clusters, and there's actually 20 tissues here, so there should be in fact more clusters in there. So the way people work on these is that they usually do a broad clustering. For example, they find the T cells or specific type of cells, and then they remove all the other clusters and then do another round of tSNE and clustering there to find details of those cell populations and cell communities. So that takes a lot of time, and it takes a lot of expertise for people, and then finding the markers for them. So what in the middle, you see the NetL map. This is, I guess, a zoom of 200 on this data. You can see there is about 91 clusters right off the bat, and then you can see after finding the markers for them, they all have kind of clear markers for each of these clusters. So it really helps with time of analysis if you have large data. And here is another data set, actually 9 different samples provided by the Broad Institute. These are all PBMC samples. You have 10x Genomics version 2 and version 2, and there's a version 3. You have CellSeq2, DropSeq, InDrop, and InDropSeq1, I guess SeqWell, and SmartSeq2. And so these are batch aligned after batch alignment. And as you can see here, you have UMAP, tSNE, and NetL map. And you can see the clusters in the UMAP and tSNE. Usually the clusters kind of like always, you see they're very close to each other, kind of like drawing the line between them, among them. It's kind of like sort of approximation, and NetL map makes it easier to do that. So the same clusters in here, about 17, I believe, clusters, right? Yes, there's 19 clusters. So 19 clusters. Again, you can turn on some markers in here. I want to look at some of them. So you turn on CD3E again. So these top parts are all CD3E positive T cells. And then CCR7, that would be your memory T4 of them, because the other, if you turn on CD8, these are CD8 positive, and these are CD8 negative. If you turn on CD68, you see the myeloid cells in here. If CGR3A, which is CD16 monocyte, it only comes in here, as well as in natural killer cells as well, expressed at high. But among the myeloid lineage, this would be your also comes up a little bit higher in natural killer cells, but this would be your CD14 positive monocytes again there. No, sorry, this is not natural killer cells. This actually is natural. The top one is natural killer cells. GNLY, you see it turned on here, negative for CD8, but GNLY positive. So again, you turn on MS4A1, 3 clusters of the B cells in there, and then PPDP turns on in here. That's also, I guess, marker for platelet cells. And some other markers that I turned them on in here. And one other couple more slides I wanted to show you before doing some analysis. Joseph wanted to also show some slides. But here, this is a mass cytometry data. We wanted to use this as a data set that is very simple and not as complex. And unlike single cell, there's like 20,000, 30,000 genes. And here you have only 40 proteins or antibodies. It's easier to look at. It's a simpler data. So, and here we got 5 samples. You have, this is from this paper. There's a lot more samples, but I just took these 5 as an example. So you have B cells, CD4, CD8, gamma delta T cells, and NK cells. And if I plot them in here without clustering, just based on their conditions or the samples, you can see tSNE and UMAP do a pretty good job in terms of like separating them. And then when you look at KNetL map, each of these, you see they divide into multiple ones. So for example, this natural killer cell in the pink and purple, it turns out into 3 clusters. And then after you cluster them, there's 16 clusters. And you can see, for example, these 3 clusters in here of the natural killer cells, that would be cluster 15, 14, and 16. When you look at the markers in here for them, these natural killer cells, the last 3 ones, you can see this one of them is only positive for CD57. One of them is only positive for CD56. And one of them is negative for both of them, kind of. So it's kind of like makes sense that you see 3 clusters because KNetL map had enough resolution to be able to divide this one big into 3 distinct clusters. Again, you can play with the zoom in KNetL map and get more and less, resolution. But so again, in the B cells, you see one of them is negative for this marker, CD45, I guess RA. Again, in the gamma delta T cells, some of them are negative in here and some of them positive in here. And in the T4 and T8, you see, for example, this one is positive for CCR7. This one is positive for PD1. This one is, I don't know, CXCR3. So, each one of them you can distinguish with a marker. So that just proves that how much more detail you can get with KNetL map. In a simple data set as well. So, and again, you have sometimes trouble, data sets that it's really hard to cluster, like if you have, let's say, only dendritic cells or only T cells and only T4 cells, for example, and you want to find your Tregs and other exhausted Tregs and other forms of T4 cells. In KNetL map, using KNetL map, using UMAP and tSNE, they usually come as a big cluster, and you cannot really use that old technique of broad clustering because they're already only T4 cells. So it's harder to zoom in there with with UMAP and tSNE, whereas in KNetL map you can find distinctions. So in this one, cluster number 3 here, easily found here. Again, cluster 2 is in the corner here, but it can't find it in here in a separate distinction. Cluster 1, easy, that comes in here, comes clear in UMAP and tSNE 2. But then again, you have 8, 7, and 6. It's more clear in here than 8, 7, and 6 in here. So this is in fact a dendritic cell data. I guess Joseph has a lot better slides than I have in here because they're working a lot more in detail in this. But I just wanted to show you a few markers that they were interested. This is from a published data set. So LY60, they were interested. It comes up in cluster number 2. With better distinction and few of the markers that they were interested, I turned them on in here that comes with distinction. And this is my belief, but Joseph can elaborate on this. I was thinking maybe these are one cell type and they're in fact separated by the cell cycle. Here we did some cell cycle analysis and I saw that they're differing in terms of cell cycle. Maybe Joseph can talk more on that too.
Yeah, I could just jump in on that just to make a quick note. So I think the Ly6D point that you mentioned is very noteworthy here because even in the analysis from the original publication, they were unable to discern a population that we know should be there, basically.
Mhm.
And KNetL showed very clearly that this Ly6D population pulls out very neatly. And then the other note about the connectedness of the three clusters in the middle, that seems to be the preliminary leading hypothesis, is that they're all a very similar population that are hard to distinguish, more specifically dendritic cell progenitors that might actually be distinguished primarily by how quiescent or proliferative they are. That's what we think, but we have to do some more work to figure that out.
I know you have so much better slides because I saw in the figures yesterday you sent me. Okay. Yeah. But anyway, so but then again, so very quickly, I just wanted to just tell you how to run NetL because it's a little bit tricky. It's you have the same when you run tSNE and UMAP, usually people are using PC dimensions, like depending on their data. Usually most people using 10, but 10 or based on the data. So again, with NetL you have the same thing too. You use dimensions, like say 10 dimensions or 20 dimensions PCA, and then there's also a zoom option. And that makes it a little bit tricky for, for first timers who are running NetL a little bit trickier to run it, and a little bit also tricky to do the clustering. So I just wanted to show you how to do that so you can do it. I know a lot of people are now in NYU also using NetL, so just play with it easier and run it easier. So usually with NetL we usually use around 20 PCs. Let's say if you're usually using 10 PCs and UMAP and tSNE, we're usually using NetL 10 more. And that's because it's already has a lot more resolution from from the start. So you just kind of get not so many clusters. You wanna it's also with the 20. So the way I usually work on it I usually keep it in 20 and play with the zoom. But again, I haven't yet, with the 50 or so projects that I worked on, haven't yet so much, tried to change the PCA, because usually with the 20, I always got better results. But then again, you can play with that. But so the zoom is important. So if you put this around 20, and the zoom works like this, if you have lower number of cells, you usually, let's say if you have less than 5,000 cells, a zoom of around 120 is a good place to start. Again, you can zoom in and out more if you want to see more of the B cells subpopulations, or if you don't want to see more subpopulations, zoom out. So that's about the number to start. So again, if you have 5,000 to 10,000, 200 to 300 zoom is good. 10,000 to 30,000, 200, 240, and then it goes higher with higher number of cells. And that also depends on the type of the data you have. If you have a lot more dropouts and low coverage, that also affects it. So it's not always that you have to, if you have less than 5,000 cells, you say, oh, I'm going to use 100 first, and you get it maybe not a very good result. Again, you have to play with that a little bit more to adjust it. And I'll tell you how to adjust it too after you do the NetL map. So
Reza, can I ask a quick question? Mm-hmm. When you showed that kind of like a network of clusters about 10 slides ago, was that, oh yeah,
go back,
forward?
Oh, I'm getting it here. Oh.
No, it was when you showed the, yeah, right here at the bottom. Yeah, right. Yeah, right here in the bottom right. Is that? I guess what I imagine is that one can sort of intuit that if they loop through the different zooms, right? Because you would see the clusters branch out. Is that how the algorithm?
So yes, good question. So by the way, these two are using a completely different algorithms. So this one is in fact is a what they call a abstract network. In fact, in here you're trying to, so how it works is that you're trying to find so you're trying to draw lines between each of the cells to the closest cells to them, right? And then you count the number of lines that go from one cluster to another cluster, and then you just like So you only have one line, and that line, the thickness of the line and the length of the line shows you how many number of lines went to that cluster. So technically it says, okay, if you're doing Euclidean distance, right? So from this cell, what are the 10 closest cells to itself? It's mostly coming from the cells in the same cluster. But sometimes, there is a line that goes from one to the other, right? If you're not using KNetL map, if you're using, let's say, tSNE, right? Sometimes there's a line that goes from one cluster to the other. So, and then you count those number of lines, and then you just like abstract them. You just remove the lines, you only keep one line, and that would be the sum of all the other lines.
Okay, thanks.
Okay. So it's kind of like, Euclidean distance, the trees that people have. It's kind of like sort of similar idea. Okay. In fact, there's a tool called PAGA that they actually, I got the idea from them. In fact, they had abstract networks, but that's totally different than KNetL maps, method. So anyway, we just wanted to have them to just show the global structure side by side. So here we are. So we run KNetL map. Let's say you get this dimensionality reduction. There's 0 clusters. We haven't clustered them. So, and then you run clustering. And then here we have 10 clusters. And then again, I should change this K. In fact, it's sensitivity, not K. So you can increase the sensitivity in here to 50, 100, whatever, to see more. And, you have, less or more clusters as well. Just like KNetL map, you can change the sensitivity. And we're using here, we're using iCLOS. iCLOS is very similar to Phenograph, published in 2015, which is, I think it's the most popular clustering algorithm right now, because a lot of major tools also using Phenograph now. Seurat and Monocle, and everybody's like, I guess the consensus is that it's doing better than the other ones, although there's like 30, 50 different clustering methods. So iCLOS is very similar to that Phenograph, except instead of doing Jacquard's similarity, it uses Euclidean distance. And more or less, I've done a lot of experiments. It almost like 98% they're identical results. So it's, so you could use either, if you want, you can also use Phenograph for clustering. So let's say you have the clustering done, and then what we do, the trick is, so what we do, we order the clusters. What that means is that, so for example, in here, you have the T cells all in here. You have 2, 8, 5, and I guess 9 in here. And in the left, you have 1, 4, and I guess 10. 1, 4, and 10. And you see these are mylolineage. So ordering the cluster really means just to have the numbers in a consecutive order. So for example, and you see in here, mylolineage is 3, 4, and 5. They're consecutive numbers. They're very close to each other, right? And here you see 6, 7, 9, and 10. They're much closer to each other. So the way we do that is that it's very simple. You average each cluster. So you have in here 10 columns, and then you take the top 500 genes that are expressed, highest expression of the top 500 genes, or you can change that. And then we do a Euclidean distance, and then we say, okay, this, these 10 clusters, this and this have more genes similarity in terms of gene expression. So it kind of like makes it easier to get consecutive numbers, and then it relabels them to new numbers. So then, and then it changes the numbers. And the reason we do that is that once you do that, it makes it easier. Once you find the markers, they come closer to each other for you to be able to see which clusters are more similar to each other. In this case, there's all these clusters that we found. You see this number 17 and 18 come very similar, right? Although they might not be, they might be different. So what you need to do is that you show this heat map beside the KNetL map, and then look at them side by side, and then just see, okay, how much are they in the similar, very close to each other? Are they like, like a snake, maybe, and then based on this and your expertise, you decide maybe these two need to be merged. And then you can relabel 17 to 18, and then you lose cluster number 17. And then it becomes one cluster. And then again, you do the reordering again, and that fills that lost cluster, and then do this process one more round. Again, and this can also help you to decide what zoom is better for you to use. And it's really like 2 rounds or 3 rounds of playing with this process. You can get the clustering and the zoom in a nicer way for you to like you know, have your data in a much better way. And really, a lot of people here also analyze single cell data, and I guess the consensus is the clustering is the hardest part of the single cell analysis, and sometimes it can take months. And yes, this might be like you might need to do this 3 or 4 times until you get the right zoom or something, but believe me, this would take much shorter time than like doing the broad clustering, taking all the, you know, another tSNE, doing all the T cells one more round. Or some people also do over clustering. They, let's say in the first, they get 20 cluster, and then they increase the sensitivity and they get 100 clusters. And then they again find the markers and then merge them one by one that way too. But then again, with the thing with tSNE and UMAP is that because when people do over broad clustering, maybe I can show you one more slide, they're already doing approximation. And it's not like a clear-cut line in there. And that already causes, you know, when you say, oh, I want to take all my T cells, you see your T cells already mixed with natural killer cells sometimes, you know. And then when you take your T cells away, there's some of the natural killer cells coming with it when you want to do a reclustering again on them. So with KNetL map, you don't have that problem. It would make it much easier for you to reduce the time of the whole analysis. So I guess to be quicker, let's have Joseph go and share his slides too, and then I'll do some coding as well, show you how to run it, maybe one example if you have time.
Great, I'll just share my screen now. Thank you for the thorough description, Reza. Okay, I hope everyone can see my screen. Let me put it into presenter mode. Great. Oops. There we go. Okay, so I think the purpose of what I want to do here today, and by the way, my name is Joe. For those of you that don't know me, I'm a postdoc in the Reyes lab in the Department of Pathology. I'm not a bioinformatician. I'm a wet lab scientist, immunology by training, and I've been working at length with Reza lately on some of our single cell data sets. And we've been working together since pretty early days of iCellR, and I've seen kind of KNetL come about and be applied from its inception. So what I want to share with you all today was, I guess, just some more case studies, some more examples of how to put this to use, and in my experience, how using KNetL over UMAP and tSNE has really been transformative for all of our studies. And some of the data that I'll show you today is actually being packaged together as we speak, and we're submitting a manuscript on this shortly. Okay, so the first data set that I'll jump into, and I'll try not to make this too prolonged because I know Reza wants to go through and show more examples with coding specifically, but I'll just show you some of what we've seen as the advantages. This data set was from bone marrow hematopoietic progenitors. After QC and everything, it came out to about 6,000 cells. And this is our clustering based on UMAP that shows that we have some early myeloid progenitors, some even earlier multipotent progenitors. And importantly, the cells that we were most interested in were these dendritic cell progenitors here. You can all see my mouse pointer, right? Let's do this one. Maybe this is better. Okay, so we have these early dendritic cell precursors here, or progenitors, and then we have precursors that are more strictly flavored towards the mature populations that we know: plasmacytoid, classical, and there's also some monocyte precursors here as well. But for the purpose of our study from the biological perspective, what we were truly interested in was getting a thorough analysis of these DC progenitors here. Okay, and as you can see, you know, they're fairly well separated from the rest of the early progenitors, but we were hoping that we would be able to get more structure within this population. So in the early days of our analysis, we actually were doing a lot of manual curation based on expression of different marker genes that I won't show you here right now, and expression of actually some surface markers as well, to show that there is heterogeneity in this population. Now, when we took our data set and reclustered and redid the analysis from the beginning and also reclustered using KNetL, what we found right away is that our DC prime, this is how we call them, DC prime, DC progenitors, they split up very nicely now. So here, this is the same clustering designations from the UMAP analysis now applied onto the KNetL map. And you can see very clearly that cluster 5 here splits very distinctly into 2 separate populations now. And when we look closer at this, we find that, well, nicely, what we were already hypothesizing based on our UMAP analysis has come to fruition in this unbiased way of analyzing the data using KNetL. And clearly what this has shown is that with KNetL, we've been able to, as Rez has been using this term, zoom in more on the populations we care about to get the biological importance, the biological relevance of the clustering in a more distinct way. So then what I'll also show you to really impress upon you how nicely KNetL performs on actually this data set, which I'd say is fairly challenging compared to most others. If we go back and look at tSNE, tSNE would look like this for us. Again, this is the UMAP now overlaid with clustering from, sorry, one second, I just can't see my screen. Okay. Now, if we go with the KNetL analysis and iClust, where we have clustering designations for each of the KNetL clusters, the KNetL populations, we can see that tSNE and UMAP do a fairly poor job of separating all these populations that we found quite a few of them are biologically relevant distinctions that are being made here. Importantly, I really would like to stress that these other algorithms were unable to really tease apart these differences in such an obvious way that KNetL was able to. Okay. Now, just jump into one other data set that we've been working a lot with. This one is a bit more complex. It's also bone marrow. It's also hematopoietic progenitors and precursors from the bone marrow, isolated in a slightly different way that I think for our discussion is not terribly important. But there are many more cells. Now we're up to about 18,000 cells that passed QC. And I think what's also very important here is that there's a wider range of phenotype in this data set, and there's also more potential for subtle variations within smaller populations. So again, one of the goals of this study was to get a fine look with high resolution at the population of DC progenitors. And here they are, cluster 6, again, blue, conveniently. And you can see that UMAP does a fairly good job of separating out all these populations that are very different from each other. But again, the DC prime cluster here is not really showing much structure. We take the same data set, same cluster designations, and now just put on the overlay this on the KNetL map. We can actually see that, first of all, 6 has this unique structure here. But there's also cells from cluster 6 that are now way up here, and are interacting and associating with other cells from the CDC megacluster from the UMAP analysis. You can also see very clearly that a lot of these other clusters have split up into different populations, and we've actually been pretty astounded at how much more biological context we can pull out from this data set in such a rapid fashion. To put it into context, we had spent about probably almost 10 to 12 months already with using tSNE and UMAP approaches. And when we jumped into KNetL, it's been about maybe just a couple of months that we've been using KNetL, and we've really increased how much useful information we can pull out of this data set, I don't know, tenfold.
So Joe, I have a couple of questions.
Sure.
One, we need to be clear about what is our evidence that these subclusters are real.
Yeah.
And I'm sure you guys are working on it. On top of that, from a more technical angle, I think what we need to do is maybe follow the standard approach, overclustering using UMAP, or taking these subsets from the UMAP and reclustering them, and really understand if the new subclustering based on UMAP, it's cumbersome, as Reza is saying. Yes, we know that using KNetL has advantages, but at least we want to show that if we follow that standard approach, we get similar subclusters. I mean, actually, whatever we get is going to be interesting. If we get the same subclusters, our argument will be that we simplify the process, and that's fair. If we get different subclusters, then we need to understand this a little bit better.
I see. So the question is whether if we try to get higher resolution using UMAP or tSNE, do we get the same answers we get with KNetL?
Exactly, and we can do this both ways, in two different ways at least. One is overclustering from the very beginning. The second one is do two-tier clustering, where you do a broad clustering, then you take the monocytes alone and you cluster them alone.
Yeah,
I think it's a very interesting question, and again, whatever the answer is, I think it's going to be cool to see.
Yeah, and that's a fantastic recommendation. Thank you, Aris. It's actually part of what I was saying about how much time we had spent with UMAP was exactly in this process. We had done multiple iterations of the UMAP analysis, and even before that with tSNE. Although the most extensive analyses we had done were with UMAP. And what we found was that even if we try to get better resolution on the entire data set, we could never do better than this, first off. And also, when we honed in our analysis on just cluster 6, for example, or even on this half of all of the populations, we still couldn't break up our cluster of interest into more refined, biologically relevant populations. And we...
Perfect. So Joe, that makes total sense. For the purpose of the paper, I think we just need to find a nice way to demonstrate it. And when we talk about biological relevance, we need to have some kind of metric that says, look, these subclusters have no markers that make sense, for example.
Yeah, you need to show evidence, Joseph.
Yeah, of course. No, we have, trust me, we have plenty of evidence. I didn't want to take up too much time talking about the biology of the data set per se, at least for this forum. I mainly wanted to just explain the advantage.
Do you have some markers for each one of these clusters to show in next slides, maybe?
In this presentation, no. I was focusing more on just the clustering itself, not on the biology behind it. But if anyone's interested, for sure, we can dive in more. We have plenty of analyses here to back up everything I'm saying.
I guess I saw the figures in the manuscript last night, and I was like, I thought you were going to present those.
No, I didn't want to get too much into it because the biology of this is a bit esoteric, I think, for this forum. So I didn't want to dive too much into that. But like I said, if we're interested, we can
discuss. Actually, maybe ask the group if they would be interested to see maybe a follow-up of this discussion with real biologically relevant arguments as to why KNetL is potentially useful and better. And we can do a little poll, Itai, maybe after we're done. to ask people
Yeah.
if they're interested.
Yeah, that sounds great.
Sure, we could do that, of course.
Yeah. Okay. Yeah, I guess for now, you have to just kind of take me for my word about the biological side of things. But yeah, as Aris has said, if anyone is interested, we certainly could do a deeper dive into all things dendritic cell, and I'd be happy to participate in that.
So can I ask a question?
Sure.
So when you say, I don't understand the biological side. So have you used CytSeq? So you have used antibody markers to verify some of the data, or no?
Yes. Yeah. Both of these data
Oh, you
sets,
have. Okay.
yeah, are CytSeq data sets, actually. Yes, you're right.
Okay, so they are CytSeq data sets. So it looks like then you can use this analysis potentially instead of using CytSeq. Is that true?
Oh, no. So to clarify, the CytSeq is the technical approach to generate the data. And then what we're talking about now is understanding the best way, the best algorithm
Oh,
to
I'm
use.
sorry.
But you're including the antibody data in, so CytSeq includes antibodies. So this data includes
Right.
antibodies and RNA. Is that what you're saying?
Yeah, so actually the clustering
Oh.
itself though is based solely on the RNA transcripts. We don't use the antibody tags for clustering purposes. For one, because it can be redundant. If you have the RNA, the transcript data and the antibody tag, it will make it put twice as much weight on those markers. And we've seen generally that the RNAs, well, the ADTs can perform quite well as also, but we generally stick with just the RNAs because there's more information there. For example, in understanding the heterogeneity in our DC progenitors, most of that heterogeneity is not captured by our surface markers.
Right, but I think that the question that I would like to know is the biological relevance. So is there anything that came out of this analysis indicating that these subgroups that were not seen in UMAP or that were not seen in other clustering mechanisms have a functional distinction?
Okay, so the functional.
We don't have to do it now, but I think it's, to me, it's like, looking at abstract art until it has a biological meaning.
Sure, yeah, and I totally get
think, as I said as I texted to my postdoc, the cluster that you pointed to looks really weird. It looks like a hookah pipe to me. So,
Oh, yeah, this one, right?
Yeah, or I don't know if it's a hookah pipe or a coiled cobra, but it's like a bunch of Rorschachs until you show us a biological output.
Yeah, and again, as I mentioned, we had limited time here, so I didn't think we had enough time to dive in all the way on the dendritic cell.
Just a few examples, just one or two examples would be helpful.
Yeah, and we can definitely do that for sure. If people are interested, I would be happy to do that. I guess what I can say for what relevant discoveries come out of this. So the focus of our paper was to understand the path of dendritic cell development, and especially one of the major focuses of the paper is understanding if dendritic cell development is primed at earlier stages, even before, and if there's priming within the DC progenitor population. You could maybe refer to this as like the CDP population for those familiar with the field. If there is signs that there are individual cells in the CDP pool that are specifically primed to go into the PDC lineage direction or the CDC1 or CDC2 directions. Okay, so what we found was that there are clear distinctions, there are clear signs of heterogeneity in this population, and that because we had the CytSeq data, the antibody tags there as well, we found that there are actually a couple of markers that were potentially useful in identifying these cells prospectively. And we're working on showing the functional significance of these and showing that these markers may in fact be able to be used to prospectively identify individual CDPs that are primes to go into only one direction. The paper also has a lot of other information with clonal tracing that really supports the overall story. So this isn't the entire story, but like I said, it's quite a long story, and I can show you more data for sure. I just don't want to take up all of our time today. So, okay.
So, Reza, maybe at this time you can use the last 3 minutes to sort of wrap up.
Yeah, it's only a couple of minutes. So yeah,
in my share in here.
So I don't think we can run any examples, but if anybody was interested to run an example, just go to the iCellR website, and there's a few examples. There's also, so if you need the code, and this one has all the code, and if you run these codes, you should get exactly similar result, identical results as you've seen here. So this one has a batch align analysis. This one has if you actually go back in the page, there's few. So there's the mouse cell atlas in here, full cytometry data in here. There's one, a couple more examples in here. We'll put more and more in there, but the first example is also in here. The pipeline, so you can just copy paste the codes from here. You should get identical results as in this page. So yeah, I guess to be quick, yeah, you just run the NetL map, and then you decide on the zoom and how much detail you want to see, and then kind of do the clustering. Also do the clustering based on NetL map because it just comes more precise in that way. So I don't, because we can't code it anymore, So this is my 2 cents on this. I'll do it. Is there any question? It's also freely available here in iCellR and CRAN. If you go to the R, where is it? The link. If you go, you can download it from R through install packages iCellR.
Well, maybe one thing we could do is have another single cell club session where we could do 2 things. One, pick up on where we were at about the biological issues, and Joe could have more time because we are interested. And then 2, Reza, maybe you could give us a kind of tutorial where we all download the data and we all have our R open and execute your code and see that we can use this.
Sure, yeah. I'm happy to do that next month, or next second 2 months from now, or whatever.
Sure, whenever works.
Yeah, and also iCellR is, yeah, I'm happy to do that. So
I think, guys, it's a good idea to do it next time in November, so that people don't forget. There's no reason to wait.
Mm-hmm.
This.
Yeah.
Push in our minds. Great. And if other folks here have ideas for additional topics to discuss, you can write to Aris, you can write to me or to Kim, and we're very happy. We're always looking for volunteers to present. This is your club, and like I said at the beginning, feel free to use it for your research. So, Aris, would you like to conclude?
Yes, I mean, the only thing I want to say is this is a beautiful club, and we really enjoy being together. Hopefully, in a couple of months, we can really be together.
Yeah, that would be great.
To get to that point. But yeah, it's been a great club so far. Thank you for everybody who's contributed presentations and questions. Thank you, Reza and Joe, for today, and looking forward to the next one in November. Bye, everyone.
Great, thanks.
Thank you, Reza. Have a great day, guys.
Do we have time for questions?
I have